So it's a pleasure to introduce Kildare Miranda, who is Director of the Advanced Microscopy Unit and Vice Director of Senabio. And he's going to be talking about navigating the current landscape of bioimaging at a national facility in Rio de Janeiro, dreams and reality. And I think we all have that duality of the dreams we have when we set up a facility and then the reality of actually doing it. So over to you, Kildare.
So thanks, Lucy, so much for the kind introduction. And also thanks to the organizers for the opportunity to talk about what we've been doing in Rio, in Brazil. Senabio is a national facility that was built with the dream to study a biological phenomenon from the atom, so across scales, from the atom to the whole animal. So for that, we established some infrastructure to study macromolecules by NMR and X-ray diffraction. So we have machines from 400 to 900 megahertz, a whole set of magnets. And also on the other end of the scale, we have some bioimaging infrastructure to image small animals, so bioluminescence, MRI, PET scan, echo, ultrasound. And microscopy is in between, so my talk will be focused on microscopies by the part of the Senabio that I'm responsible. And then we have two microscopies has the ability to bridge between cells, organelles, and molecules. And the other end of the scale, cells, tissues, organs, and the whole animal. So we have to develop these techniques and new technologies in the center and sometimes in the country. For that, we have a light microscopy facility with super resolution, turf, fluorescence stereo microscopy, a multiphoton instrument. We actually are replacing this one now. Also fluorescence correlation spectroscopy, optical tweezers, so a bunch of techniques to study cells in the light at microscopy level. And then we have an electron microscopy facility, so with different machines where we do from basic EM to three-dimensional reconstruction, room temperature, and also cryo with FIB-SEM or tomography. And more recently, I'm going to talk about our recent challenge to install a cryo-EM unit at Senabio, sorry. So one of the things that we're particularly focused in our center is to image or to obtain information about ions within cells. So one of the focus that we have in our center is the study of infectious disease. So we're able to map three-dimensional distribution of ions within organelles in parasites. So this is a very strong part of the center that we do. And we also are particularly interested in quantification of our three-dimensional sets. So this is something that we are pushing a lot to have quantitative data, not only nice to quantify different organelles, interaction between organelles and structure, both at light microscopy and also EM level. And then our recent challenge was to install in Rio a high-end cryo-EM facility capable of doing both single particle analysis and also tomography in cells in solution, and also tissues. And you can imagine that doing it in Rio, in Brazil, imposes some challenges. So before we were doing only basic entry-level cryo in the old instruments using side entry cryo holder. So for that, we have two instruments with CMOS cameras, but we needed more. So we reached a situation where we couldn't move forward without new instrumentation. So then we started with our cryo-EM thing. So we had to construct a new building. So you imagine that constructing a new building has a lot of challenges. See, we had received some unexpected visitors. So this was actually in our construction site. So Vlad said that we already have Python embedded. It's actually not the case. And then we finally got the instruments installed. So it's a Titan Credos G4 with the selectors filter and detector. Also, we installed the cryo FIB-SEM for cryolamellar preparation. And we already obtained our first results with the instruments. I'm really happy with that already with the correlative work in our trypanosome. So it's just a glimpse of what we've been doing. So these organelles that I showed you, that we are analyzing the elements, so we can find phase separation within these organelles. And it's really nice to see the reconstruction of this. What we see here as an electron dense content is actually polyphosphate linked to ions. And these are results from the instrument that was installed months ago. So really happy to have already some results showing this phase separation of ions within organelles. It's something that we are really excited about. So, and then going back to other things that we do at Senabio. So once we installed our center in Rio, partnered with some groups in Campinas at CNPM, which have a Titan G3. Krios also high-end cryo-EM. And the idea is to have these two centers as national hubs. So we're working in Brazil as with this hubs model, right? That where Senabio and CNPM would be other, we're helping other groups to establish their entry-level cryo-EM so that they can print the samples and ship them to do the basic work and ship them to Rio or Campinas and have high-resolution work. And so far, we have been successful in Paraná. We just received a new instrument, a new 200 kV cryo instrument at Fiocruz in Rio de Janeiro. They also just got the grant application approved. We'll have a third Titan in Campinas at CNPM, and also another one in Rio, not in my place, but at Fiocruz. So this is something that is growing, and we expect to grow also not only in Brazil, but also in America. We have this project in different countries, in Latin America and in Europe, that was approved, this EULAC project. So this is also this hubs model that we are implementing there. So to establish things, test them in Brazil, and expand to Latin America. So we also been working a lot in mapping the landscape of microscopy landscape in Brazil. So one of the things that we knew, but we have never quantified, is that there's a lot of asymmetry on the instruments in Brazil. Just electron microscopy, we have around 700 operating instruments, and most of them are concentrated in the south and southeast region of the country. And because of this, we made an effort and went to the Amazon to implement a new center there. And this is tropical diseases, on climate change, and on the sustainable use of Amazon resources. So this is something that we're excited, and this lab is part of our network. So we've been also involved with a lot of training, organizing courses. So this was kindly organized by Global Bioimaging last August. Gleb was there, and a lot of other to give a facility management workshop. We also had the first Latin America crime workshop in the fall, actually 2 weeks after this, after facility management workshop, and we also organized the LABI meeting. So I'm a co-chair of LABI, and we organized LABI in August, very busy August in Rio de Janeiro, where we met a lot of the friends that we see here. It was really great to have them in Rio. So lessons learned. So I talked about our dreams, the things that we, but now we have to deal with the problem that creates. So one thing is that it never ends, right? Never ends. So new technologies are always arising. It requires more funding, doesn't people, more problems, more solutions. It never ends. It will never end, right? We have people to do that. So collaborative work is essential, but it can be a very lonely process. It's very hard to get more in this collaborative work to work for the community and not for your own project. It's something, but it's it's because they really want to, right? Unconditional support from the unconditional support from the community. This happened already, so the grants are approved. We have a lot of visibility to the fund chips for technicians. And then what we need now, we need computational power software people. From my perspective, we're only able to do that if we work with the community. So thank you. Right on time. Community, community, community. I think that's what we should do. So with that, I would just like to thank my team and there, and also thank the funders. Arigato. Thank you.